Okay, today's topic is based on how throughout the term we investigate different aspects of a specific genetic disorder. Disorder I had was the propionic acidemia, and basically I had to prepare at least a minimum of 10 slides with a description of the genetic disorder and the major characteristic, molecular cause of the disorder, mode of inheritance, simple pedigree illustrating the inheritance pattern, options for genetic testing, and including a reference. So, that being said, let's start this presentation right um, right now. So my topic today is on propionic acidemia. Official name and etc. is PCCB, Homo sapiens propional CoA carboxylase subunit beta, transcript variant 1 mRNA. Gene symbol is P05166 PCCB underscore human. History. Around the early to mid 1950s, this disorder was found within a patient who had a deteriorating mental development, multiple attacks of acidosis, and high levels of ketones and glycine located in the blood. In 1961, the Johns Hopkins Hospital, it was clarified that the disorder was a recessive disorder and was originally called ketotic hyperglycemia, which is the same as propionic acidemia. It is noted that in 1970, a male Pakistani's offspring of two first cousin parents had led to that child's death at age 8. Description of this disorder: propionic acidemia is when the brain and nervous system are affected by this disorder. Occurs a few days after birth. The symptoms are a lack of appetite, vomiting, weak muscles, and lack of energy. The progress issues of the disorder often lead to heart abnormalities, seizures, coma, and death. Can have a delayed development as a child, and it also does not have the ability to fast to longer periods of time without food. So you always need constant nutrition. Continued more statistics on this is 1 out of 100,000 people, based off the quality health professional in the United States, are affected by this disorder. This genetic disorder is most commonly found in parts of the Inuit population of Greenland, a few Amish communities, and also in certain groups from the Middle East culture, who often have intertwined family marital status. Gene and chromosome location. Mutations located in the PCCA and PCCB genes cause a mutation of the enzyme called propionyl CoA carboxylase. This enzyme helps in the breakdown of proteins. This mutation causes there to be a shift in the breakdown of protein molecules and can affect the brain and nervous system. Inheritance. This is an autosomal recessive disorder that has two copies of genes in a cell that both have a mutation. It is an autosomal recessive disorder, which illustrates that both parents have to be carriers of the gene. Genetic testing. This can be identified by newborn screening, which measures the levels of certain metabolites such as propionate and methionine in a blood sample, and molecular genetic testing as well. Prenatal screening, amniotic testing, chorionic villus sampling, and amniocentesis, another form of testing. Testing mandatory. Testing is based off the diagnostics that are done on a newborn the first few weeks of their lives. Detailed history of the patients and also the family history is also checked as well. It is usually found out before the birth or even shortly after. Where is the testing done? Shortly after the birth of the infant. During this time, the first few weeks of the newborn is there are heavy testing done to make sure that it is not an issue with a newborn's health in any way, and also to make sure that not any prenatal testing occurs before birth as well as the baby progresses in its development. Samples from the fetus or uterus during pregnancy. The typical cost of this testing can range from $100 to no more than $2,000, which is based off the degree of testing. Costs also increase based on if the person needs more than one testing or if multiple family members need testing as well. Newborn screenings vary state by state from a range of $15 to $60 per infant. Molecular causes, summarizing it, basically it's based off two subunits of the biotin-dependent propionyl CoA carboxylase, which is a mitochondrial enzyme that is involved in the catabolism of odd fatty acid chains. This branch chain of amino acids, isoleucine, threonine, methionine, valine, propionyl-CoA carboxylase catalyzes the carboxylation of propanyl-CoA/propionyl-CoA to D-methylmalonyl-CoA, methylmalonyl-CoA being similar. And with the holoenzyme, the alpha subunit catalyzes the ATP dependent carboxylation of the biotin carried by the biotin carboxyl carrier (BCC) domain. The beta subunit then transfers the carboxyl group from carboxylate biotin to propionyl-CoA. Propionyl-CoA carboxylase also acts on butyryl-CoA, which is converted to ethylmalonyl-CoA. Ethyl-CoA being similar. Other alternative minor substrates include the 2-E-butynyl-CoA, crotinyl-CoAs being similar, and is used processing normal proteins. Carries out a specific way to break down several protein building blocks. Also breaks down lipids and cholesterol. Implications: there are more than 100 mutations involved with this gene, PCCB. Mutations are on the cytogenetic location, as shown. And the changes in the single DNA building blocks, nucleotides, and inserts or deletes genetic material in the PCCB gene. PCCB gene causes the prevention of the product of phenyl-CoA. This causes the change or missing part of the enzyme or protein from being broken down correctly. This is a sample pedigree chart. Propanoacidemia is a autosomal recessive disorder. On the right and the left are the parental genes at the top, which are homozygous recessive, little a, homozygous recessive, little a for both parents. And it shows that the cross between these two parents has the offspring F1 generation to have 100%, the disorder probability of having 100% of the disorder, and this simply shows that trend of autosomal recessive behavior displayed in this cross. And then these are the citations. Thank you for listening to our presentation today. And that snoring sound is my little Frenchie right here. Say hi.